Good morning to all. Hello to everyone joining today's webinar. I'm Dr. Pradeep Gulani, and as the co-convener of this event, I'm delighted to welcome a diverse and engaged audience. Your participation greatly enriches our discussions and underscores our shared commitment to advancing in our respective fields. I look forward to a productive conversations and collective learning as we embark on this educational journey together. SPSR is distinguished for its engaging webinars, further enhancing its status as a leading platform for continuous professional development in pharmaceutical sector. I'm confident that today's session will uphold our tradition of excellence, providing valuable insights from a respected speaker. Let us seize this chance to gain new knowledge, participate actively, and progress our field. Welcome, everyone. I look forward to a productive experience for all participants. Thank you for joining us today. We are gathered today to commemorate the 87th SPSR webinar on plant-derived anti-epileptic agents, mechanism of action, and therapeutic potentials, a distinguished event hosted by the Society of Pharmaceutical Sciences and Research. Under the esteemed guidance of Dr. Shashi Alok, the National President of SPSR, the Society continues to play a pivotal role in fostering innovation and advancement in the pharmaceutical sciences. The SPSR is devoted to advancing the pharmaceutical field through a variety of strategies. Our mission is to organize conferences that support pharmacy-related initiatives and endorse programs tackling public health issues in India, focusing particularly on disease prevention. We make it a priority to recognize and honor pharmacists whose notable contributions drive progress in our field. At the heart of our mission is a commitment to spreading pharmaceutical knowledge, ensuring a steady and robust exchange of information about pharmaceutical practices and services. By striving towards these objectives, we aim to elevate the quality and impact of pharmacy profession, fostering a proactive and collaborative community dedicated to excellence. It is our great privilege to introduce Dr. Damanpreet Singh, Principal Scientist, Pharmacology and Toxicology Laboratory, in charge Zebrafish Research Facility, CSIR-Institute of Himalayan Bioresource Technology, Bioresource Technology, Palampur, Himachal Pradesh, India, as a distinguished speaker. Dr. Damanpreet Singh expertise greatly contributes to our event, providing substantial knowledge and insights. His participation is certain to enrich our discussions and expand exploration of plant-derived anti-epileptic agents. We are grateful for his engagement and anticipate an enlightening session. It is our privilege to introduce Mrs. Monika Sabharwal as the convener for today's webinar, who also serves as the founder and National Secretary of SPSR and CEO of SPSR Periodicals Private Limited. Her significant involvement and leadership in the pharmaceutical sciences and research communities are indicative of her commitment to the field. We are very grateful to have her guidance in today's session. I am pleased to invite Mrs. Monika Sabharwal to warmly welcome our distinguished guest speaker. Over to you, ma'am.
Thank you so much, Dr. Pradeep Golani. Hello, everyone. I am Monika Sabharwal, the convener of 87th SPSR webinar. I extend a warm, heartfelt welcome to all of you. We are thrilled to host this enlightening session on plant-derived antiepileptic agents, mechanism of action, and therapeutic potential, organized by Society of Pharmaceutical Sciences and Research (SPSR). We are extremely honored to have with us today distinguished and inspiring speaker, Dr. Damanpreet Singh Sir, Principal Scientist, Pharmacology and Toxicology Laboratory and in charge of Zebrafish Research Facility at CSIR-Institute of Himalayan Bioresource Technology, Palampur, Himachal Pradesh, India. Sir, your contribution to the pharmacological research, especially in the domain of natural product based therapeutic, is truly commendable, and we are eager to learn from you insights today. This webinar had received an overwhelming response, with more than 1,700 registrations from over 20 countries, a true reflection of global reach and the impact of SPSR platform. We are grateful for your continued support, and your enthusiasm encourages us to raise the bar with every session we organize. As we gather today to celebrate the 86 successful webinars behind us, I take this moment to appreciate the tremendous support we have received from the global pharmaceutical and medical community. We are constantly motivated by your encouragement, and which we have received through the various platforms every time. Your words fuel our efforts and strengthens our motive. At SPSR, we are committed to uplift the pharmacy profession by conducting free, knowledge-rich webinars on diverse and relevant topics, many of which are suggested by you, our audience. Our webinars bring together experts from academia as well as industries, promoting collaboration, conversation, and the knowledge exchange across various sectors. We are also excited to announce the upcoming 5-day Faculty Development Program FDP titled Strengthening Teaching, Learning and Evaluation Process in the Higher Education, in collaboration with School of Pharmacy, Sri Shankara Professional University, Bhilai, Chhattisgarh, and supported by IIT Bhilai. This is another step in our journey to empower the educators and enrich academic practices. SPSR continues to work hand in hand with the leading pharmaceutical institutions to organize national and international conferences. And the response this year has been especially heartening. Many institutions are now expressing their willingness to collaborate with us, and we look forward to more partnerships in the future. Our SPSR YouTube channel has grown rapidly, and now proudly it has over 21,000 subscribers from more than 50 countries. According to the feedback we received, the more than 98% of the participants feel that the SPSR webinars have contributed significantly to their professional development. That is humbling and motivating our achievement for all of us. We are also thrilled to witness a rapid growth of our SPSR membership community. Participants from the pharmaceutical and the healthcare backgrounds are actively joining hands with us, helping us to strengthen our network. If you wish to become a member, please visit our official SPSR website. We also welcome the proposals for the institution collaborations and MOUs to further share our goals. Through this journey, we ensure that all the participants receive their participation certificates, which are valuable additions to their professional portfolios. The fact that we are able to offer this entirely free of cost makes our initiative even more exclusive and impactful, especially for the students, researchers, and professionals worldwide. We invite all of you to continue sharing your topic and the speaker suggestions so we can make every webinar even more relevant and impactful. Our webinars, hosted every Sunday, are steadily building a tradition of routine knowledge sharing that will benefit all of us. Before we commence, I would like to warmly welcome our National President, Dr. Shashi Alot. whose visionary leadership has been crucial in the impressive transformation of the SPSR. I would like to extend my heartfelt thanks for the significant contributions made by Dr. Pradeep Golani, the co-convener of this webinar, and organizing committee members, Dr. Saurabh Kosek, Dr. Sanjay Nagdev, Mr. Ankur Agarwal, Mr. Junaid Tantre, and Mr. Rishabh Bhanot. Their tireless efforts have played a crucial role in transforming this event into a reality. Join SPSR community of over 50,000 pharma professionals actively engaging across LinkedIn, YouTube, Facebook, and WhatsApp. Let's connect, learn, and grow with the top tier webinars and the exclusive content every time. Do not miss out on this unparalleled opportunity to elevate your pharmacy practice. Stay updated by subscribing to SPSR YouTube channel and enabling the notification bell icon for the instant alerts. Follow our journey in advancing the pharmaceutical sciences that will benefit the society. Once again, I extend my deepest gratitude to the today's speaker, Dr. Damanpreet Singh sir, and to all of you for being part of ever-growing SPSR community. Together, let us continue to striving to expand the knowledge, empower professionals, and uplift the pharmaceutical sciences community worldwide. Within that, let's begin today's scientific exploration. Thank you and welcome to the 87th SPSR webinar. Thank you so much and over to you, Dr. Pradeep Golani.
Thank you, ma'am. I appreciate Mrs. Monika Sabharwal granting me the opportunity to present this webinar. And now let's begin with the short introduction to guest profile. Dr. Damanpreet Singh is a distinguished pharmacologist and researcher currently serving as the principal scientist at the CSIR-Institute of Himalayan Bioresource Technology, Palampur, India, where he also holds the additional responsibility of coordinator, Academy of Scientific and Innovative Research, an institution of national importance. Dr. Singh completed his m.pharm in pharmacology from Guru Nanak Dev University, Amritsar, followed by a PhD in pharmacology from Punjabi University, Patiala. He began his academic career as an assistant professor in the Department of Pharmaceutical Sciences and Drug Research at Punjabi University before transitioning into scientific research at CSIR-ISBT, where he has since risen through the ranks to become a senior and then principal scientist. He has also served as a visiting scientist at the National Research Institute of Chinese Medicine, Taipei, under the Ministry of Health and Welfare, Taiwan, further enriching his international research exposure. Dr. Singh's research is primarily focused in the field of epileptology and neuropharmacology, with a strong emphasis on the development of natural product-based therapies for the management of epilepsy. He is credited with establishing the Zebrafish research facility at CSIR-ISBT, which has become a valuable platform for neurobehavioral research. His scientific excellence has been recognized with several prestigious awards, including the P.C. Dandiya Award, Dr. Hardayal Award, and Saroj B.N. Sharma Award from the Indian Pharmacological Society, as well as the Early Career Research Award by SERB, DST, Government of India. Dr. Singh has also played a pivotal role as a co-nodal officer for the nutraceutical and nutritional missions under CSIR New Delhi. He is an active member of various scientific societies, including the Indian Pharmacological Society, Indian Academy of Neuroscience, and the American Academy of Neurology. He has also published numerous peer-reviewed research articles, filed patents, and serves on the editorial boards and reviewer panels of several reputed international journals. Dr. Singh continues to contribute significantly to the translational pharmacological research bridging natural products discovery with neurotherapeutics. It is with great satisfaction and pride that we declare that we have exceeded the 1,700 registrants globally, showcasing an impressive diversity of countries. Significant attendance is noted from nations including India, Algeria, Egypt, Ethiopia, Indonesia, Iraq, Kenya, Malaysia, Mexico, Nepal, Nigeria, Oman, Pakistan, Saudi Arabia, Sri Lanka, UAE, and Uzbekistan. We are deeply moved by the substantial and varied responses we have garnered. Our heartfelt gratitude is extended to each person who has taken a moment to participate in this significant event. Thank you all for joining us today. Let's make this webinar an insightful and enriching experience for everyone involved. We have our esteemed keynote speaker, Dr. Damanpreet Singh, sir. Welcome, sir. Your insights will be highly valuable, and I would appreciate if you could present them in a speech. Over to you, sir.
Thank you, Dr. Pradeep, for such an elaborative introduction. And I'm also very happy to listen the goal of your society toward betterment and upliftment of pharmacy profession. So before I start my talk, I would like to thank the President, Dr. Shashi Alok, the convener, Monika Sabharwal, and you, Dr. Pradeep, for giving me this platform to interact with such a huge global audience from different nations as well as from India. So not taking much more time, if you permit, can I share my presentation?
Right, sir, you can share, sir.
Is it
Right, sir, it's visible, sir.
okay? Okay. So as Dr. Pradeep have already mentioned the title of my talk, that is plant-derived anti-epileptic agents, mechanism of action and therapeutic potential. So before I start, I just want to introduce to the audience our institute where I'm working. So you can see this is the snapshot of the entry of our institute, that is Institute of Himalayan Bioresource Technology. Second one, you can see our laboratory, and third, it is the thing for which this place is very much famous. It's a tea, because it is also called the Darjeeling of North India, the Palampur. So as far as our institute, it is an institute under Council of Scientific and Industrial Research, that is an autonomous body of Ministry of Science and Technology, Government of India. It is the only institute that is situated in Himachal Pradesh. And if we see about whole CSIR, it has 37 national laboratories all over India, 39 outreach centers, 3 innovation complexes, and 5 units. And it is the only lab that is situated at Himachal Pradesh under the foothills of Dholadhar range of Himalayas. And this is the whole activity of our institute. If we see our mission, it is developing technology for boosting bioeconomy through sustainable utilization of Himalayan bioresources. And at our institute, we work on different domains to go for product development and focusing on translational research. And our core area is on industrial crops, aromatic plants, medicinal plants, floriculture, food and nutraceuticals, bacteria, fungi, algae, and development of diversified products. And for all of this, we are wholly focused on working on Himalayas. So now if I say about my topic, that is epilepsy. So it is a disease, I would say it is a disorder, because there is a little bit confusion what exactly it is. Either it is disorder or it is disease. But as per new definition, it is a disease. And it is a chronic neurological condition that is manifested by abnormal production of electrical impulses. This is what we just say it is epilepsy. There is just an abnormal production of neuronal discharge. Sometimes it appears in a form of seizure, sometimes it remains as an absence. So depending upon the type of seizure, we classify it into different ways. I think the names of the countries Dr. Pradeep has taken, majority are developing countries. And if we see the load of epilepsy, it is more on developing countries. Around 80% of the cases globally, they resides in developing countries. And globally, there are around 50 million persons that suffer from epilepsy globally. I won't say that this is an exact number, because in individual nation the definition of epilepsy gets changed, and because of this change in definition, the cases that are reported are very less compared to the true number. But still, as per WHO, there are around 50 million people that suffer from epilepsy. And in spite of availability of more than 30 types of anti-epileptic drugs, one third of the cases remain drug resistant, that they won't get cured with the available pharmacotherapies for epilepsy, that making the condition more and more worse, that the current therapy is not fulfilling the need of individual patients. So if we see the availability of cases at urban and rural population, it is 64% in urban and 35% in rural area. This won't say that the rural area, the prevalence is less. It is that the reports of epileptic cases are less in case of rural areas. So these are some of the major types of epilepsies that appear in clinical situation. So if we go for the etiological factors, depending upon the age, the etiological factors it get changed. During infancy and childhood, the major causes are prenatal or birth injuries or inborn errors of metabolism, congenital malformation. Childhood and adolescence, it is majority, sometimes there is some genetic defect that later on developed during the time of growth, or it is a CNS infection, either it's malaria, tuberculosis, schistosomiasis, neurocystosarcosis, or meningoencephalitis, trauma. In adolescents and young adults, there can be a traumatic injury that is a major cause, and drug addiction and withdrawal of drugs, abrupt withdrawal of drugs, sometimes are the core reasons for development of epilepsy. And in older adults, major it is stroke, brain tumors, and certain type of other condition. But in spite of this, around we say 70 to 80% of cases are idiopathic. We don't know the exact cause. So this is a condition of epilepsy. But if we say it is not only epilepsy that affects the quality of life, because if we see epilepsy, it is if we go with this pathophysiology, it is very much complex. It is not just that the neuron it starts to seize. There is an abnormal depolarization. It's not only this thing that is happening. Once this neuron it start abnormal depolarization, it affects the adjacent neurons. And the areas of the bodies that are controlled by that particular region, they also get affected. And that's why the epilepsy it's not only become the single condition, it become the complex neuronal condition that affect other body parts. And the condition that appear, they are called as epileptic comorbidities. If it is these comorbidities are limited to the brain only, then we get the condition like psychiatric disorders. There is a high prevalence of anxiety disorders, mood disorders, and several other types of psychiatric condition in epileptic patients. There are other somatic disorders. There can be stroke, migraine, reproductive dysfunctions, cardiovascular diseases. I have highlighted these cardiovascular conditions because I have we did something on this condition that I will explain later on during my presentation. So this is just that all other organs, they are also affected. And if we say about available anti-epileptic drugs, they hardly affect this condition. And in some cases, they they itself worsen these conditions. Like the person that are taking anti-epileptic drugs, apart from getting cure of epileptic seizures, they get more they get more prone to these conditions itself, just because of the nature or the mechanism of the drug where they are acting. Apart from these two, there are some cognitive disorders that are usually appear during or they usually appear in epileptic patients. There are some sleep disturbances, disabilities, accidents, nutritional problem. And the major challenge for the treating these conditions along with epilepsy, it is a complex pathogenic association. Like I said that cognitive disorders. So we know that during cognitive disorders, we want the brain should get excited. There should be some more glutamatergic activation. And if we go for the increased glutamatergic activation, there is a more chances of worsening the seizures. So that's why treating such diseases or such conditions simultaneously with epilepsy it becomes challenge. For the management of this, there is the need of multi-targeted therapies. And as you all know that plants, they are the cocktail of multiple bioactive metabolites. That's become ideal to treat such type of complex condition where there is a need of multiple targeted approach. And moreover, the available therapies, they either target one or two pathological markers of the disease. That's become difficult to treat such conditions simultaneously. So there is a need of comprehensive management therapies for a better control of epileptic seizures as well as for these all comorbid conditions. So I just want to show you schematically that what exactly it happened during epilepsy, how the drug targets, and what are the needs that we want to have to establish a better therapy for epilepsy patients. So if you see, this is a normal condition. If we see these are the brain functions, both excitation and inhibition. Excitation, I just say it is a depolarization that happens whenever there is any response inside the brain or the brain want to do any function. And inhibition, it is a state where there is maintenance of, we say, resting membrane potential. And normally we say in the brain, excitation and inhibition is very precisely controlled by different neurotransmitter functions. We say excitation through glutamatergic mechanisms, inhibition through GABAergic mechanisms. So these are precisely controlled inside the normal brain. And what happened during epilepsy? There is an abnormal excitation or there is abnormal downward shift of inhibitory functions. So there is excessive depolarization inside the brain. And this is the outcome of epilepsy, that there is excessive depolarization. So the red thing that I have shown, that is the root cause. Something it happens inside the brain and there is an abnormal excitation. And if what available therapies do, so this is where the conventional drug therapy act. Either they inhibit the excessive excitation or they promote maximum inhibition so that the depolarization, the abnormal discharges of the brain get inhibited. But if we see what exactly it is required, we want a therapy that should target this red one, the root cause that is relating to the abnormal excitation. And all the available drug therapy we see, so these are the pathways, these are the drugs that are currently in use. Either they act on sodium or potassium channels, the sodium or calcium channel or potassium channels, just to inhibit the depolarization or to prevent the propagation of electrical impulses, or they act on GABAergic mechanism to increase the GABAergic tone, or they act on the glutamatergic mechanism to inhibit the excitatory tone. No one the drug is targeting on the neuronal changes that are leading to such type of abnormalities. So we try to work on this area. We searched out several literature that what happened inside the brain that ultimately lead to depolarization, excessive neuronal discharge. So we just try to correlate what happened during epilepsy before going for identifying the root cause. So if we say all type of epilepsy, not only we say temporal lobe epilepsy, in majority of the cases, there is an establishment of epileptic foci in the brain. So this foci can be established by any of the etiological factors that I have already explained in my earlier slides. Either there is an injury or there is some genetic abnormality that is creating this epileptic foci. And after that, there is a latent period. Like if you see during any traumatic injury, there is an injury and ultimately in majority of cases after some time there is appearance of recurrent seizure. Sometimes there is seizures at the time of injury also, but majority of cases there is appearance of continuous seizure after some latent period. So what happened during this latent period, there is some changes that happen inside the brain. There are some abnormal things that get established inside the brain. There is either hippocampal lesions that get established. There are reorganization of neuronal circuits. There are some mossy fibers sprouting. That this fiber they sprout out, and there are some other neuropathological conditions. And after this latent period, these changes has been happened. Then there is spontaneous recurrent seizures. Then there is a symptomatic epilepsy, that epileptic discharge that we see in a patient. And all of the available drug therapy, they target on this pathway. They target on spontaneous recurrent seizure in any way, either through ion channel, either through excitatory function or through inhibitory function. They target on spontaneous recurrent seizure. But if we see there is a need of a therapy that can prevent the changes that are happening inside the brain that are leading to appearance of spontaneous recurrent seizure. So this green area is a place where we can have where we can target to have a better therapy to prevent the appearance of epileptic discharge. So we try to find out what are the pathways that are responsible that act on these abnormal discharges. So we try to make changes, and we found the literature from different sources, either from the whole genomics, whole proteomics, transcriptomic studies that are available, and we try to merge all these in a single pathway. And if you can see, all the changes have been shown, like if there is an injury, there is an inflammation, and there is activation of interleukins, and something it happens. There is excessive glutamate production, glutamatergic activation, then there is some pathway that are interacting. So by compiling all these things, we found that this green area that I have shown, it is mTOR. It is mammalian target of rapamycin. We get the hyperactivation of this pathway that play a crucial role in development of all these pathological changes that happen during that your latent period after development of epileptic foci. So in any case, if we can target this mammalian target of rapamycin, we can have prevention of appearance of seizures. So this was our assumption that this can be done. And there are initially some literature was available that shows that this mTOR play a crucial role in certain type of genetic epilepsies, or condition like we say there is a TSC mutation. This is again a genetic disorder where there is appearance of epileptic discharge. So we found that this can be a potential target, and that can be targeted using some natural products. So before proceeding further, we try to validate this target. We use a known inhibitor. Pardon?
Uh, Sir, your slides are not moving, sir.
Uh,
Please check it, sir. Right,
so
sir. Make
I
it.
am
You can make it a full screen, sir.
no, it's fine.
Now it's fine, sir.
Okay, so this is the pathway for which I was discussing, that we merged several pathways to make this thing, and here we found that this mammalian target of rapamycin is an ideal where we can have something. And before proceeding further, it was necessary to validate this. We use a known inhibitor. We use LY294002. It is commercially available inhibitor of an upstream target, that is PI3K, that is phosphatidyl inositol 3 kinase. Is a kinase inhibitor. By inhibiting this, there was downstream inhibition of mammalian target of rapamycin. So to prove this, we use fish model. Actually, these pure molecules, these are very costly. And testing this in rodent model, it become high. It demand a lot of money. So that's why we establish a zebrafish larva model, where we treated this larva with this inhibitor, and then we excited the brain to see that whether these changes are happening and whether there is inhibition of mammalian target of rapamycin. So we use this fish model. You can see when we expose this fish to a chemoconvulsant, this larva, there is a hyperactivity. A hyperactivity is just because of excessive muscular twitch that we get in the larva. So you can see I have shown distance traveled and average speed as a marker of hyperactivity. I just want to show you this video. So you can see here there is some whirlpool-like movement. The larva is rotating. And this condition, it mimic a little bit generalized seizure that we appear in case of human patients. And if you see here, you can see these are the clonic seizure, the lower one, the purple color. You can see the larva, it has shown these are the clonic like seizures. And these were our endpoints. And by observing these endpoint, we concluded that there was suppression of seizure after inhibition of PI3K. And ultimately, it was showing the downstream inhibition of your mammalian target of rapamycin. And this is how we tested in adult zebrafish model. Here we injected intraperitoneally, and we note down the severity in a similar way. So it just give an idea that if we can target that mammalian target of rapamycin, we can prevent the production of abnormal changes that are happening inside the brain. To go further, uh, we were required to make some active molecules that target PI3K. So for this, we collaborated with our chemistry group led by Dr. Praledas, and we have an bioinformatics group led by Dr. Rituraj Purohit. So we requested them to make some molecules for us. So they selected deodar tree because, as I have shown in my initial slide, that our aim at IHBT is to use the Himalayan bioresource. And these deodar tree, they are present in abundance at this place. And from these plants, our chemistry group, they isolated a starting material that was alpha, beta, gamma himachalins. And they get the source from deodar oil. And then they make some semi-synthetic derivatives of these particular molecules. And then by the help of our bioinformatics group, they docked, they screen, and they try to find out some potential molecules that can inhibit PI3K, and ultimately it can down-regulate the mammalian target of rapamycin to prevent seizures. And they make a lot number of molecules, then they go for screening using bioinformatics tool, and ultimately they come out with these two coated molecule PBS8 and PBS9 to be the potential one that have a tendency to be developed as a drug for better management of epilepsy. So we tested in again zebrafish model, and then we go for molecular expression studies, that whether the whole pathway that I've shown earlier get down regulated or not after treatment with these molecules. So you can see it has shown the down regulation of particularly this mammalian target of rapamycin. And this CFOS that I've shown, it is an marker of seizures in zebrafish larva, and it was down regulated. That clearly showed at molecular level that there was suppression of epileptic seizures after the treatment of these semisynthetic derivatives. And it was published, and now it has been patented. Patent has been filed for these compounds in majority of the countries, and now we are in a process for further development this as a potential molecule for the treatment. So another compound that we have tested, like I have shown in my earlier slides also, that epilepsy is not itself where we want the treatment. We want a comprehensive management of epilepsy as well associated neurobehavioral conditions. So now to expand our work to check whether this target it has some potential for the management of epilepsy associated conditions also, then we screen out all the bioresources that are available with us. And we found that a plant that is Ginkgo biloba, actually in India we have abundant amount of Ginkgo biloba that is grown at CSIR-IHBT. So we selected the leaf of this plant. I think you all are very well aware this plant is a best remedy for neurodegeneration. And we tried to find out something out of this. No doubt it contained a toxin molecule that itself known to be a pro-epileptic agent, that is ginkgotoxin. But we standardize our extraction process in such a way that the ginkgotoxin it get removed from that. So we focused on ginkgoflavonoids, and we use the leaves, and then we go for the characterization, and then we make a condition similar to that of human, where we can get epileptic seizures that appear spontaneously, as well as there is an production of epilepsy associated comorbidities. So what we did, we used scopolamine to inhibit some peripheral effects of pilocarpine that is known to excite the brain. So we injected pilocarpine to rats and we allowed the animal to show status epilepticus. And that status epilepticus, it induced an epileptic foci inside the brain. And then we allow the animal to have a latent period. And during that latent period, we start giving our ginkgo biloba extract. Not extract, it was a fraction. And then we tested the severity and frequency of epileptic seizures. So you can see GB50 and GB100. 50, it is a 50 milligram dose, and 100, it is a 100 milligram dose. The severity was reduced, but there was no change in, we say statistically, there was no dose dependency, but the seizure severity did get reduced. And then we tested the anxiety-like behavior, the animal that were injected with ginkgo biloba with epilepsy, they showed lesser lesser behavioral alterations. So mean they have a tendency that they can be this extract, it can be further developed for the use of epileptic patients where there is appearance of anxiety disorder along with seizures. Moreover, it also improved the cognitive function. We tested the special memory function using Morris water maze. So you can see there was some protection in the special memory function of epileptic patients, epileptic animals that were treated with ginkgo biloba extract. And then we tested what were the effect on the changes that happened during the latent period in the brain. So you can see we have targeted on neuronal count, the dark neuron that clearly indicate the degenerative neuron. They were lesser in case of the animal that were treated with ginkgo biloba extract. Moreover, the mossy fiber sprouting that have shown during the process of epileptogenesis, my previous slides, it was reduced in dentate gyrus region and CA3 region of the hippocampus. That were the main reason why we were getting the protection in epilepsy-associated cognitive deficits. And at molecular level, we found that there was downregulation in mammalian target of rapamycin at genomic levels as well as at proteomic level. So this is IHC. So we also tested the glutamate level. So what we concluded that this pilocarpine, when we have injected it induces status epilepticus, and during latent period there was excessive production of glutamate, and due to glutamate there was excitotoxicity that at longer duration it induces spontaneous recurrent seizure, as it has reduced the level of glutamate also inside the brain. And moreover, the molecular changes that we tried to establish, they were suppressed after treatment with ginkgo biloba. But here we won't get any protection toward epilepsy associated depression like behavior. Means ginkgo biloba, it was showing protection against epilepsy, it was showing a protection against epilepsy associated behavioral abnormalities and cognitive deficit, but it showed no protection toward epilepsy associated depression like behavior. Then we moved further towards some other molecules of our interest. So we focus on crocin. That is a we say it is a carotenoid that is present inside the Crocus sativus. Actually, our institute it is working on promoting the cultivation of Crocus sativus, that is saffron, in non-conventional area. Like it is grown in India, it is grown in Kashmir. And our effort with the efforts of our institute, we are now started its cultivation at Himachal Pradesh also. So it was a crop of our interest. We use crocin from this plant because it has known to alter the neurotransmitter functions, several death processes of neurons. It has shown alterations and several other protective effects that supported that we should use this crocin for this particular condition. Here we use different model. Here we use pentylene tetrazole induced kindling model because our work was on pure molecule. We want a model where there is a use of lesser amount of a test drug. So what we observe that when we go for the treatment of animals with an epileptogenic agent along with crocin, it reduces the severity of seizures. Moreover, it prevented appearance of epilepsy-associated cognitive deficit. When we studied it on T-maze, it shows protection against special memory functions, as well as it prevented your recognition memory on NORT, that is novel object recognition test. It showed the protection, but the things were same, that it also won't show any protection against epilepsy-associated depressive conditions. Here, what we found that it reduced the reactive oxygen species content. It improved the antioxidant defense mechanism inside the brain. Moreover, it reduced the inflammatory response that is induced by reactive oxygen species in the form of downregulation of NF-kappa B and its activated phosphorylated form, that is phospho NF-kappa B. And we found that it has some potential, and this work it was selected as the best paper published in from India by Indian Pharmacological Society during their conference in 2017-18, and it was awarded by Dr. Hardayal Award by Indian Pharmacological Society during its annual conference. So then we moved further, and we focused on some flavonoid molecules. As I have already shown that our interest at our institute is to use industrial products for development of value-added products. So here we have used Apigenin because it is one of the best flavonoids available in citrus plants. And as you all know that Himachal, it is known to be as a food basket of India. So we have a lot of citrus fruits that grow at this place, and they are used at industries level. And the end product is of use, but the byproducts that are produced after the use of the citrus fruit, particularly peel, it go waste. And the Apigenin, it is present in abundance in the peel of the citrus plant. That's why we use this Apigenin, that whether we can have some product out of this. So it showed interest. We got interesting responses. The Apigenin failed to protect seizures, so it was a negative finding. It showed no protection in preventing the epileptic seizures in case of animals that were subjected to pentylene tetrazole as a convalescent. But it prevented seizure-associated cognitive deficits. So overall, what we concluded that it also prevented depressive-like behavior. It also prevented depression-like behavior. It also prevented epilepsy-associated cognitive deficits. At the same time, it increased the 5-hydroxytryptamine level inside the hippocampus. And we found that it was overall increasing the neurotrophic functions inside the brain, but it was showing no effect against epileptic seizure. So it has a potential not to be established as a anti-epileptic, but as an adjuvant therapy. So what we concluded that it is happening like this. Epigenin increased the 5-HT level that prevented the downregulation of CREB, and by virtue of which it improved the brain-derived neurotrophic functions, and by virtue of which it prevented the epilepsy-associated comorbid central conditions. So this is how we proceeded further. So as of now, what the slide that I have presented, we have focused on epilepsy and epilepsy-associated cognitive or epilepsy-associated neurobehavioral comorbidities. But if we see in epilepsy, there is another condition, and that is serious. That is one of the most serious condition, or the one of the most serious outcome of epilepsy. That is SUDAP. That is sudden unexpected death of epilepsy patient. The person die, and there is no clue why the patient has died. And moreover, this is a normal death. The post-mortem studies are very less. How to find out what was the cause of death? But there is a death. That's why it is called as sudden unexpected death. So one interesting finding that we found while working on these models, then we try to explore it. We found the application of this particular target in some other condition also. So one of my student, Arindam, he was working with the mammalian target of ripamycin, and he was working on an agent that is mycophenolate mofetil. It is basically it is used in case of organ rejection. So we found that it prevents the upregulation of mammalian target of ripamycin inside brain also. There was some trial that was done with some infants where they have used mycophenolate mofetil to control the seizures. So we were working on this, and he come to me with an interesting finding that, sir, when I go for dissection of the animal after epilepsy, because we have to transfuse some particular strain inside the body to get to have a better view of the brain. So we use transcardial perfusion. So he said that, sir, when I go for dissection, the heart of an epileptic patient is not epileptic animal is not is a little bit disoriented. So when we isolated it, we found that the heart of epileptic animal, it was little bit elongated. So we try to correlate what exactly it is happening. No doubt it was not a part of our interest, but still we try to find out what exactly it is happening. So during the time of development of epilepsy, we note down the changes in mean arterial pressure. We note down the heart rate after sacrifice. We noted the QT interval on electrocardiography of the animal. We found that during latent period, there was reduced in blood pressure. And after that, once there is an spontaneous recurrent seizure, the pressure get increased and increased and increased it further. So it was a quite interesting work, quite interesting finding to work upon. So then we planned a thorough study. What we did, we again injected pilocarpine. During the time of spontaneous recurrent seizure, we note down the seizure frequency, we note down the cardiovascular function, and at the end of the study, we remove the heart and we go for the whole transcriptome studies of that heart. And simultaneously, we perform the whole proteomic studies. We use Illumina NovaSeq 6000 system for transcriptomic analysis to see what are the differential genomic changes that are happening inside the heart, so that the plant material that we have established can have some use for this condition also, to avoid death in epilepsy patient. So after getting the findings of transcriptomic analysis, we go for QRT PCR analysis to validate the findings. And then we use proteomic finding. Then we follow Western blotting to validate that proteomic outcome. So you can see this is the whole summarized result of total proteomic analysis. So you can see this NAVG, these are the normal animal. Apg, these are the epileptic animals. So it was quite interesting that inside the heart of an epileptic animal, there was around 1157 differentially expressed genes. It means these genes were not normal. Either they were overexpressed or they were down regulated in epileptic animals. And when we go for the pathway analysis, and we found that these are the pathways that have shown to be get altered after the epileptic discharge. And then we make a network that all whether we can make whether we can use all those 1157 genes to make a pathway that what exactly it is happening. So you can see there are some orange and there are some pink target that has been shown. Orange, they are intramodular, and pink, they are intermodular. Pink means these are the target of a single pathway, and orange, these are the target of multiple pathways. And these are the crucial one, that if any of this target is removed, the whole pathway will disrupt and the condition will not appear. Means if we can target any one of this, then the SUDAP condition, the damage of heart will get reduced, and there is a chance that we can prevent a person that is suffering from epilepsy to avoid death. So these were just an initial results, and then we go for proteomics. That sometimes it happens that the things what are happening at genomic level, at mRNA level, they won't get translated at protein level. So simultaneously, we also performed proteomic analysis, and at proteomic level, we found that there was around 1264 differentially expressed proteins that are altered in the heart of an epileptic animal. So we performed, so here we make a relation that this blue color, these are the genes that are getting differentially expressed at proteomic mRNA level. Green, they are the proteins that are affecting at proteomic level. And this pink color, it is showing that these are the major targets that are affected at gene level and protein level. I mean, these can be targeted to prevent this condition. So we performed QRT-PCR analysis that validated that the finding that we get at transcriptome level, it is happening at mRNA level. These are the Western blots that show that what is happening at proteome level, they are affected in true at the wet lab findings. And then we make an finding that these are some of the things that get affected, and the outcome was published in a highly reputed journal. It is Epilepsy, and this is the official journal of International League Against Epilepsy. So this work was having a lot of importance because it gives several targets that can be targeted to avoid a death of an epileptic patient because of cardiovascular irregularities. And what we concluded that during seizures, not only the central nervous system, autonomic functions, they also get altered. And we say the respiration as well as the cardiac changes, cardiovascular system that is under the influence of autonomic nervous system. And because of this autonomic alteration, there are the certain changes that we get at when we performed blood pressure, electrocardiography, and molecular changes. They totally support that. So now our interest was to find out that what can be done by using natural molecules, that whether we can target something or not. So it was that the first, and in the same outcome, what we found that again the mammalian target of ripamycin was very important. Here also, if it can be down-regulated in the heart also, it can prevent the degeneration or the damage of the heart as a consequence of continuous seizures. So to validate this, there was a need of an animal model because I have already told that the pure inhibitors, they are very costly. Sometimes the cost is even more than the cost of total project. So that's why there was a need of a small model where we can test our hypothesis. So first we developed a new model of zebrafish where there is an appearance of epilepsy is same as that of human. So we developed a kindling model, and in this kindling model, we used a known inhibitor that was rapamycin. That is a known inhibitor of mammalian target of rapamycin, and we found that when we treated the fish having chronic epilepsy with this inhibitor, there was suppression of cardiac damage. So we have correctly validated our findings. Then we try to search out some plant that has known to be used for cardiac irregularity as well as it is used for epilepsy. So we found that Nardus jetamansi. It's also called as mansi. So its rhizomes are used in traditional medicine also. So we selected that, we performed extraction, we go for UHPLC, Qt of MSMS characterization, and we characterized or we identified these 15 molecules. And then we perform the docking with these molecules to that target that we have validated or that we have identified from our whole transcriptome analysis inside the heart. So they showed a very good interaction with that particular targets. And then we tested this compound in rat model. We, we induced seizures in rat. Then we treated those rat with the characterized extract. Then we studied the whole molecular changes inside the heart. And we found that the target that what we identified at transcriptome level, they were suppressed by the treatment of Nardus jetamansi. And the damage in the heart was very less compared to the untreated epileptic rats. So it has some potential to be developed further. So the whole work that I have presented were performed by all of my these PhD students. Arindam, he worked on mammalian target of ribamycin to validate their efficacy and to identify their protective effect in epilepsy as well as epilepsy associated comorbid condition. Supriya, she worked on epilepsy as well as cardiac irregularities, the work that I have shown. Pallavi, she worked on Apigenin, and Savita, she's presently working uh, uh, for her PhD, and she worked on development of a new model of epilepsy. So this was the outcome of all these students, and this is my group, and these are my contact details. If any one of you have certain queries and you want more information on the work that I have performed, I will be very happy to link with you. So you can find out more about our lab from our personal lab link. So I have shown my Twitter. This is my mobile number. It's also on WhatsApp. So you can contact me at any time. So this is a tulip garden at IHBT. Uh, Earlier we used to find tulip at Kashmir, but now from past 3 4 years we had tulip garden at IHBT also. And I welcome you all. If anybody want to visit, it will be open now at next year, because for this year it has been closed. Thank you.
Thank you so much, sir, for
Thank you.
delivering us such a brilliant and insightful lecture on plant-derived anti-epileptic agents, mechanism of action, and therapeutic potentials.
So if
Your
anybody
deep expertise. Yes, sir. Yes, sir.
if anybody have any query, I love to answer.
Yes, sir. I will be asking the queries from
Sure.
our participants after this. So, deep expertise and engaging presentation gave us a comprehensive understanding of how natural products can contribute to epilepsy management through innovative and mechanistic approaches. We truly appreciate the clarity with which you explain complex pharmacological concepts and your contributions to advancing the neuropharmacology using models like the Zebrafish. Your session was both informative and inspiring. Thank you once again for your valuable time and remarkable contribution to our 87th SPSR webinar.
Thank you,
So
doctor.
now, sir, I will be asking the questions asked by our participants. So the first one is, sir, how do natural compounds compare with conventional anti-epileptic drugs in terms of targeting ion channels or neurotransmitter systems?
Actually, the work that has been performed using natural products toward these targets, they are not thoroughly studied very well. If we see that natural product where we have targeted or the available literature is available that used a cocktail of compounds. So in a single extract, you say there are hundreds of molecules that they target on different sites. And in many of the cases where some studies have been performed using the specific target, they have shown equipotent response. Like there are several, you say sorelin, bergaptan type molecule, they have shown a very well target toward GABA aminotransferase in a similar manner as that of conventional drug. But there is a lot of scope to work in this area where you can use a specific isolated molecule and test it on those targets that have been well studied using conventional drugs.
Right, sir. Thank you so much, sir. The second one is, sir, are there specific plant constituents known to modulate the neuroinflammation or oxidative stress in epilepsy models?
Yes, definitely. Two I have already shown. Crocin, it has shown a very good response. You say nuclear factor kappa B, it has shown inhibition of that. And moreover, ginkgo biloba that we have tested, it has shown the reduction of interleukins that are the main inflammatory mediators. And apart from what we have performed, there are several other compounds that have shown suppression of inflammation that is induced by continuous seizures. There are a lot number of molecules.
Right, sir. Thank you so much, sir.
Thank
The
you.
next one is, sir, what are the toxicity or safety limitations commonly associated with natural anti-epileptic compounds?
Again, I will say that this is the area where there is need of more exploration. Toxicity is there. Like I have seen, I said that even in ginkgo biloba where we worked, there is a compound that is called a ginkotoxin that is known to be as pro-epileptic, that is known to induce neurotoxicities. So you have to take care. But definitely, I will say that not much more literature is available related to toxicity. And it become more important when you work on a specific pure molecule. The chances to get the toxicity is more.
Right, sir. So the last one, sir, what role can AI or machine learning play in screening and predicting the anti-epileptic potential of plant-based models?
Yes, that is what it is a need of the hour. because now we have very good analytical tools where we can go for screening of each and every molecule. No need to go for isolation, because we have lot number of databases where there is known compounds, their structure, their molecular weight is already present. We have to just take an extract and we note down, or to go for identification of all those compounds, and then use that information using the AI tools, because we have a structure of protein, we have good docking tools that are available, where we can
Right,
use
sir.
all this information and to go for the target. Like you can target on inflammatory pathways, you can target on ion channels, you can target on neurotransmitter function, and based upon that data, collectively using different tools, you can find
Yes.
out the best mechanism. And when you have a known pathogenesis pathway, you can use that information to develop a better therapeutic agents.
Right, sir. Thank you so much, Dr. Damanpreet Singh, sir, for answering all the questions so wonderfully and with such clarity. Your responses have truly deepened our understanding of the complex mechanism and therapeutic potential of the plant-derived anti-epileptic agents. We sincerely appreciate the time, thought, and expertise you shared with us during the interactive session. It was truly enriching, and we are grateful for your valuable contribution to our 87th SPSR webinar. So, here we come to the conclusion of this session. So, as we conclude the 87th SPSR webinar, we extend our heartfelt gratitude to Dr. Damanpreet Singh sir for his remarkable and enlightening session on plant-derived anti-epileptic agents, mechanism of action, and the therapeutic potentials. Today's presentation offered us a deep dive into the neuropharmacological potential of natural products, highlighting their mechanism of action, therapeutic relevance, and future prospects in the management of epilepsy. The discussion around zebrafish models, neurotransmitter modulation, and the role of the phytochemicals in neuroinflammation and oxidative stress has been informative and inspiring. Dr. Singh's expertise and research contributions have provided a new perspective on integrating traditional knowledge with modern pharmacology to explore safer and more effective anti-epileptic therapies. On behalf of SPSR team, we thank all the participants for their enthusiastic engagement, and we once again express our sincere appreciation to Dr. Damanpreet Singh for his valuable time and contribution. We are deeply thankful to Dr. Shashi Alok, the National President of SPSR, for honoring us with his presence. We recognize and appreciate your commitment and guidance, and we offer our profound thanks for your significant input. We would also like to express our appreciation to our convener, Mrs. Monika Sabharwal, for her pivotal role in organizing this event. We extend our gratitude to all SPSR team members for their collaborative efforts, which have broadened our understanding and fostered cooperative learning. Your collective contributions have made this webinar a success, and we look forward to continued collaboration in advancing pharmaceutical sciences and research. We look forward to your participation in future SPSR webinars as we continue to explore groundbreaking topics and foster knowledge sharing in pharmaceutical sciences. Thank you once again, and we wish you all the success in your future endeavors. Have a wonderful day ahead. Thank you, sir, from Team SPSR.
Thank you, Dr. Pradeep, for your nice words. And at the same time, I thank each and every participant for listening my talk. And I'm sure that within 50 minutes or 60 minutes, I cannot conclude or cannot provide each and every information. But if anyone, they need some specific information and where they think that I can be of any use, then do contact me. I will be happy to answer each and every query, and wherever you need, I can have a support. Thank you, Dr. Pradeep. Thank you, Dr. Alok. Thank you, Dr. Monika, again for providing this platform to interact with all of you. Thank you. So, Dr. Pradeep,
Thank
if
you,
you
sir.
permit, can I now disconnect?
Yes, sir. Yes, sir. We
Okay.
can disconnect, sir. Thank you so much.
Thank you.